Hello, everybody. Welcome to a new episode of the Dissenter. I'm your host, as always, Ricardo Lopes, and today I'm joined by Dr. Itai Yanai. He is founding director of the Institute for Computational Medicine at the New York University Grossman School of Medicine. He is also a professor in the Department of Biochemistry and Molecular Pharmacology there. And today we're going to talk about his book, The Society of Genes. So, Dr. Yanai, welcome to the show. It's a pleasure to have you on.
Thank you, Ricardo. It's a pleasure to be here. And I just want to note that this is a book that I wrote with my very good friend, Martin Lurcher.
Yeah, sure. Okay, so since we're going to talk about genome evolution, most of our conversation will be about that. Let me just ask you, do you think that the metaphor, or I think Richard Dawkins uses it as a metaphor of the selfish gene is scientifically useful to understand genetic evolution?
Yeah, it's a great question. So just for your listeners, I'd love to kind of tell the history of the selfish gene. It's a book that came out in 1976, just a year after I was born, but it took me some time before I got to it. And the main premise of the book is that the genes are what is important in biology. And I think that's such an important lesson because many times we biologists, we look at the organism and we try to understand what is the organism? What are these parts? What does this do for the organism? What does that do for the organism? And what Richard Dawkins told us is that that's really the wrong way to think about it. The right way to think about it is that the organism, the individual, is really what he called a survival machine. And what he meant was that the genes have built this individual, and the goal is just for this machine, the survival machine, to make a new copy of the genes so they can go to the next generation. And what he really meant is that the genes are immortal, while the organism is very transient, right? We each open our eyes, we live our lives over just a few decades, and then we have to close our eyes, as Richard Dawkins said. And the genes don't do that, right? When did the genes open their eyes? They opened their eyes, so to speak, 4 billion years ago. So the genes are immortal. The genes are what he called the replicators. And what we are just a sort of way for the genes to make more copies of themselves. And so I think that's a profound understanding of what's happening in biology. And the way he kind of captured that concept is by calling the genes selfish. So what he meant by selfishness is that the gene doesn't really care about the individual person. He doesn't care about us. The genes are not there to serve us, but rather they're selfish in the sense that they only care about themselves. They only care about getting to the next generation. So to answer your question, I think it's a brilliant metaphor. I think it's profound and helpful. The only problem with it is that it gets very misunderstood very often.
In what ways?
I could just tell you, when I first heard about the book, I was just an undergraduate student interested in very different things. And my first impression upon hearing the title, The Selfish Gene, is that we have genes in our body that make us selfish. And that, of course, is not at all what he meant. It's not that the genes make us a particular way. It's that we kind of use this anthropomorphism by adding to the genes a kind of quality of selfishness, which, of course, Richard Dawkins didn't mean. Richard Dawkins didn't mean that the genes have a sort of consciousness and that the genes are thinking and are trying to be selfish. What he meant is though the genes act as though they are selfish. And he has said, I read on Wikipedia, that he regrets calling the book The Selfish Gene. He wishes he had called it The Immortal Gene. But I think he doesn't mean it. Uh, Martin and I have discussed this many times, and we think the selfish gene was great as a title because it's so provocative. And for all its disadvantages of creating misunderstandings, it did draw so many people, including myself and Martin, into biology because it made biology so fascinating.
Right. So just to go through a couple more questions regarding the basics of genome evolution, is it really true that mutations occur randomly, or, I mean, is it possible that at least in certain circumstances they might follow patterns?
Ah, that's a great question. So mutations do follow patterns. Uh, in some cases, I'll tell you about one exception. Mutations are actually, in a really crazy way, learned from the environment. That's through CRISPR, and I'll tell you about that in a second. But uh, the the basic thing to realize is that mutations are, in a very fundamental way, random. And while you might think, well, if mutations are random, how can we get something like a frog or a fish? You know, what can randomness ever create something so complex? And I think that's a really big sort of obstacle for people to overcome in order to understand how evolution works. And the basic thing to understand is that the fact that mutations are essentially random uh, is is um, is not a detriment to the theory because there's a second part, and that is selection. So it's as though you're talking to someone and you're trying to decide what should we do tonight, and your friend is giving you all kinds of ideas. Well, we can go to the karaoke bar, we can go to see a play, we can go hear some jazz. They're giving you all kinds of ideas, and maybe they're all random. And then you may be the selector. You may say, "Oh, I think it's best to go hear some jazz tonight." So it's this two-part process that's also happening in evolution. Mutations are just naturally occurring, but then there's a selection step, and that selection step is very much non-random. And so then you asked about patterns. There are patterns. There are kinds of mutations that happen more than others. An A will tend to mutate more to a T than to a C, and a C more to a G than to an A. So there are these patterns, but that's still, you know, within these sort of constraints, there's still a tremendous amount of randomness. And I'll tell you about one example that's really the exception to the rule, where mutations are not random, and that is CRISPR. Because, and I'm talking about CRISPR not in the way that we've all heard it in the news as such a great method for gene editing, not for the reason that it was awarded the Nobel Prize, but just I want to remind us that CRISPR was not invented by humans. It was actually invented by bacteria billions of years ago. And the function of CRISPR is to act as an immune system. What happens is a virus will attack the bacterium, and the bacterium, if it's lucky enough to fight off the virus and to beat it, it will keep a little record, a little kind of snippet of information, and then it will integrate that into its own genome. So that's a mutation. It's now mutated its own genome, but in not a random way at all, in the least random way possible. It's actually keeping a memento of the virus. And the way the system is worked, this system called CRISPR, is that these little mementos now serve to fight off future infections from the very same virus. The bacterium is now constantly searching its own self for evidence that the same kind of sequence of a virus is now attacking it. So that's a case where the mutation to the genome is not random at all. But that's a really unique case. And 99.99999% of all the other cases of mutations that we know of are effectively random.
Mhm. I understand. So, in evolution, do we find lots of gene transfer between species?
Right. Yes, we find so much gene transfer that it has even called into question whether it's possible to figure out the family tree of bacteria at all. What's happening is that uh, while humans, for example, we have a genome, we actually don't have one genome, we have 7 billion genomes, right? For each of us, and we're all different, but all these polymorphisms together account for sort of one genome, or as Martin and I like to refer to it, as kind of one society of genes. And the way it is in our species, the society of genes is very blocked off. There's like a wall, and it prevents most other genes from other species to come in. But in bacteria, it's totally different. In bacteria, there's very little to kind of block off a transfer of one gene from one species to now integrate itself into another species. And this is happening in a rampant way, which uh, Martin and I have described as one of the great sort of pieces of evidence for the selfish gene theory, because look at how evolution really is happening at the level of the genes, right? The fact that they're jumping from one species to the other really just shows how they run the show. Anyway, if you were to sort of make what's called a phylogenetic profile of a gene, you would see so much evidence for jumping around. And what this phylogenetic profile is imagine you make a list of all the species that you know about, right? Actually, we know about millions of bacteria species. And in each species, you just put a 1 or a 0. Do we see a copy of that gene or not? And many times they've also mutated by now, but you could still sort of infer that it is the same gene. So now you make this profile of 1, 0, 0, 1, 1, 1, because it was in this species, this species, this species, 0, 0, because it was not in these two species. So you make this long vector, right? We call it the phylogenetic profile. And what you see is that this vector is so spotty, it's so random, and the opposite of what you would expect if the genes were just inherited from sort of mother to daughter. Because if genes were only inherited, then you would see a profile where it's just 1, 1, 1, 1, 1, 1, 1, because all these species belong together, and then 0, 0, 0, 0, because these are all very distant species. But you don't see it. You see a 1, 0, 0, 1, 0, 0 craziness documenting this rampant jumping around. So it seems that the only set of genes that don't really jump around are what we call the informational processing genes. These are genes involved in translating a mRNA, kind of like converting the nucleic acids into protein, all these ribosomes. These don't, for some reason, transfer from organism to organism. And so when we try to make a family tree of the species, we use these informational processing genes, because everything else is just crazy jumping around.
Right. And since you mentioned the Society of Genes, could you tell us what that is exactly? I mean, do you use it as a metaphor or do you use the term society literally in the sense that perhaps biologists would use it to understand animal societies, for example, and the kinds of relationships conspecifics establish among themselves?
Ah, yeah, that's a great question. So we don't use the word society literally in the sense that, of course, we don't imagine genes as kind of forming a parliament, literally, or meeting to establish a constitution. It's not this kind of society. We use it as an analogy, and the notion is that the genes that we have and which form a genome are um, it's not a preordained sort of list. It's a bunch of genes that are actually forming a really complex set of relationships among them. Some of the genes are actually competing with one another. Other genes are cooperating. There are maybe classes of cooperating genes that working as complexes, and I'm sure we'll get to soon. And there's a lot of conflict. And so while we might uh, think of the genome as kind of like a simple instruction manual or a blueprint, that's not really the case. Because our genome is the product of evolution, it has all of the what you would expect from an evolutionary process. And you know what? It's kind of similar to I guess the change that you have to make in understanding the genome as a blueprint to the society of genes that we have in mind is the same kind of change that you would have to make in thinking about nature or like, a forest. So, if you went to Google and you searched nature and then clicked on images, you would find these beautiful images of like a sunset and like maybe a cute puppy and all these animals running around free. And that's the image that some people have of what nature is. Of course, the truth is when you go into a forest is that actually that's not nature at all. Nature is, as the poet once called, red in tooth and claw, in the sense that there's a lot of fighting, that the animals are eating each other, and it can be very violent and much less cooperative than we think, and more of a competition between species. And so the same thing is happening in the genome. The genes have formed this sort of very tenuous union where there's a lot of competition and as well as cooperation. And so the way Martin and I think about it is that whereas Richard Dawkins described the gene as selfish, he was so right in that vision that it now needs to be extrapolated more to the genome, right? The genome is many, many selfish genes. And it's very interesting to think about what are the strategies that the genes can take. What happens when a new gene comes in and it's now in direct competition with another gene? What should be the strategy of that gene? And it may seem as though we're committing a anthropocentric error by saying that a gene has a particular strategy. Of course, the gene doesn't have consciousness, so how could it have a strategy? What we mean is, though, if you look at the dynamics, it acts as though it has a strategy. And by inferring that strategy, you really learn a lot about the dynamics of evolution. So it's really useful as an analogy.
Mhm. But in the context of being part of the same organism, I mean, in what ways do genes compete with one another? Because I would imagine that if there was lots of conflict, then perhaps they would undermine the entire enterprise of being part of the same organism, and perhaps the chances of being transmitted to the next generation.
Right. Um, well, think about it. Let me tell you a story about lactose intolerance. So we have a gene called lactase, and it breaks down milk. And so when we are receiving milk from our mother as a baby, we turn on this gene, and it breaks down the milk, and it helps us to get all the nutrients, the sugars from the milk. Uh, and then if you came in a time machine back 20,000 years ago, you would see that after we're babies, we stop making this protein. The gene is turned off because we no longer receive breast milk. We've grown up. We don't need to receive breast milk from our mother, so we turn it off. What happened then is that our ancestors discovered that if they have a cow, they can get more milk, and that's and that's good. It's a great way to get food is now to milk the cows. The problem is we turned off our lactase. So how can we use the milk, right? Uh, and so randomly, one person, going back to our earlier discussion, this was not a mutation that was sort of generated, but just because each of us is different, each of us comes into the world with a fresh set of mutations. It just so happened that one of us, about 10,000 years ago, had that mutation, which said, you know what? Why do we need to turn off lactase? Let's just keep it on. We'll just keep turning this protein on, and we'll be able to constantly digest milk. And the person who had that now had an advantage. The thing is, though, if you look at now that situation where there's a population and one of us has this mutation, there's now a kind of competition at the level of the population between the old gene, the gene that says, you know what, we make lactase when we're babies and then we turn it off, and the new gene. The new gene says, you know what, we're going to turn on lactase and we're going to keep it on the whole time we're alive. And you really kind of see a competition. And what happens is, in some populations, one gene won. So if we look at a lot of populations in Western countries, for example, the United States, most people have the gene that says keep it on. But if you go to the Far East, most people actually have the mutation that says turn it off, and they can't digest lactose. And so it's this kind of competition that we're talking about.
Yeah, I understand. So, and since we have phenomena like pleiotropy, where the same gene contributes to different traits or its products participate in different functions, and we also have polygenic traits, that is, a trait is the product of different genes. I mean, what does that tell us about how genes work and perhaps interact among themselves?
Yeah, that's a great question. I think the first thing it tells us, the number one thing that it tells us, is that we are really the product of evolution. We really are. Uh, let me go back to how people used to think on how genes might work. So if you go back to the work of Mendel, Gregor Mendel is the father of genetics, and the kind of analogy, the kind of way of thinking that he had, and the people the scientists that came after him, was that there would be one gene and one function that follows. So you might have imagined that our genome is a set of, let's say, thousands of genes, and then each gene controls one function. So you have one gene that makes the eye. Good, I need an eye. One gene makes your hair. Good, I need hair. One gene makes your skin. Good. One gene makes my gut. One gene does this, right? And you might have expected that. Maybe had you engineered the human as opposed to evolve the human, maybe that's how you would have engineered it. But what we instead see, and this has been revealed so beautifully over the past few decades, is that exactly as you say, it's one to many. So one gene can have multiple effects. That's pleiotropy. And also one effect, for example, a nose, is not built by one gene, but by many genes. And we could talk about it all day, but I just want to summarize it in one sentence. The ultimate reason why this happens is because humans were not designed. Humans evolved. And so what had happened over the evolutionary process is that a gene might over time gain additional functions. As a new process evolves, more genes are recruited to refine it. And it's because of these changes upon changes upon changes that things get messy very quickly. And for example, we talked about the strategies of genes. So one aspect of um, the functions of genes over time, if you study what does a gene do over millions of years, we see that the older a gene is, the more functions it has. And so that leads to the notion that, if a gene was created 500 million years ago, it might have been involved in just building the nose. Although 500 million years ago, there was no such thing as a nose, but if you'll humor me for a second. But over time, it will evolve and add additional features that it's involved in. So when a gene is sort of born, when there's a new sequence that somehow is now able to create something that's going to be preserved by natural selection, it will only have like one function likely, and over time it will gain new ones. So in other words, this notion that a feature is controlled by multiple genes is exactly what you would expect from an evolutionary process.
Right. And what about the relationship between nuclear DNA and mitochondrial DNA? Is it that because they come from two different structures in the cell that those different kinds of genes get into conflict more or not?
Oh, uh, yeah. So with the mitochondria, it's crazy in one sense because they have a genome. You look at the cell, look at one of our cells, and there are many structures in there. There's the Golgi, there's the endoplasmic reticulum, there's all kinds of structures. But here is one thing called the mitochondria, and it's crazy. It has its own genome. Typically, we think of the genome as stored in the nucleus, right? That's why we call it nucleic acids, DNA, is because they're in the nucleus. So how come there's DNA now outside of the nucleus? And it's a beautiful story in realizing, and this was the discovery of Lynn Margulis, one of the great evolutionary biologists, and what she hypothesized, and she was ridiculed for this originally, but now we know from genomic evidence that the evidence is just overwhelming, that what happened is that about 2 billion years ago, what we would call society of genes, but you would call two bacteria just merged, and one became a resident inside the other. And so you have now two genomes, right? And so the mitochondria is a remnant of that original bacterium 2 billion years ago that was swallowed up. And in the beginning then, when this first happened, like the day after it happened, there was a very large genome in the mitochondria. Today we see a rather pathetic genome. It's very small, I think only about 16 genes, only like I'm guessing 60,000 base pairs, although I should check it. And my point is that it's very small and about 3 times smaller than, 3 orders of magnitude smaller than the genome of in our nucleus. And the reason is that over time, many genes transferred from the mitochondria to the nuclear genome or were just lost. And so the only genes that are still in the mitochondria, the few remaining genes, are the ones where there is an advantage that they express themselves. These are genes that are really in their core related to energy production, which is what the mitochondria is still used for today. And you ask, is there a conflict between those genes in the mitochondria and the genes in the nucleus? I would say yes, but it's not special because there's a conflict between essentially any 2 pairs of genes. Well, some genes are cooperating, but overall there's conflict. So it's no different from the 2 genes. In other words, you may be just as much in conflict with your neighbor in the building as you are with a person living in a different country.
Yeah, I would like to ask you about cancer. So, by studying cancer, can we get new insights into how genes work and perhaps the kinds of relationships they establish with other genes?
Yeah, that's a very interesting question, because a lot of times people ask the inverse question, which is, can we learn more about cancer by bringing in our understanding of evolution?
Yeah.
And because cancer is ultimately an evolutionary process. But you're asking, can um, using this occurrence of cancer, can we learn about genes? Uh, I think so. I think a lot of basic science has actually been done in the cancer field, and it's been really interesting. We learn a lot about signaling pathways, essentially because in cancer, oftentimes the signaling goes wrong. And so sometimes by studying what goes wrong, you can understand how it's normally operating. So yeah, absolutely. I think cancer is a really interesting field of research. It's sometimes what I find in doing cancer research is that one challenge constantly is to defend the doing of basic science in the framework of cancer, because there's this kind of strong notion that in studying cancer, you should be curing patients. And the opposing view is that, we should also, it's such a tough problem, this cancer problem, that we should try to also gain a really basic understanding. And like you say, also learn about basic biology in general with implications that we can't predict. So I absolutely like the spirit of your question that we should learn some basic science in studying cancer as well.
Right. So, from a genetic perspective, uh, why did or how did sex evolve? I mean, would it be would it bring any benefits to genes to be part of organisms that reproduce sexually?
Oh yeah, absolutely. Sex is this amazing invention by genes. There there could be no bigger difference than the way like we humans think about what sex is, and then the way the genes see what sex is. For so let me tell you a story, right? Bacteria uh, don't have sex as we know it. They kind of have their similar version often, but generally they don't. And so if you have a bacterium and now it has a dream, this bacterium, and the dream is to make two bacteria, two daughter cells. And each one of those has a dream. The dream is to make two. So one becomes two, becomes four, becomes eight, and it's this exponential growth that um, is really programmed at the genetic level, right? The selfish gene is replicating itself. And now let's say that one of these branches makes a mutation, has a mutation, and that mutation is actually really useful because it can now, I don't know, digest copper or something crazy. It revealed something new. And so that's a great invention. And let's say that the cousins on the other side of the tree, right, because we're making this tree, 1, 2, 4, they discovered, they also made a discovery, which is how to now take something and convert it to something else. So now these are two great inventions, right? But the best would be if you can make a bacterium that has both. But here, the way the bacteria are built, you can't. Like these guys made this mutation, these guys have this mutation, but you will never find a bacteria that has both, right? I guess it's not impossible, but you'll have to wait a long time before this clade now, where that mutation over here will happen in the background of this digesting copper mutation. So you can't kind of take the best of both worlds. sex allows you to make the best of both worlds, which is why it constantly evolves. In different species on Earth, sex will constantly evolve. It's not that it just happened once and all the animals that have sex all kind of come from that one invention. No, it keeps getting invented. It also keeps getting lost. Sometimes the genes sort of forget. They forget. They might say, why do we need this sex thing? Let's just get rid of it. But in those populations that get rid of sex, there is no future. They die out really fast. And the reason is that, so what sex allows you is to constantly mix and match, to constantly say, okay, let's make a new organism by fusing this one and this one. And so in our analogy here, there would be sort of sex, so kind of like a fusion or integration between one bacterium that has this invention, the other bacterium has that invention, and they would be able to make the best of both worlds. So that's why it's such a great trick. It really kind of allows for the genome to constantly evolve better.
Right. And since we're talking about sex, is it possible to identify a specific point where two species that come from a common ancestor split and are no longer able to reproduce between them?
Yeah. Right. So, you know, the best way actually to define a species is just as you say, that when you take an individual from this population and an individual from that population, if they can't make a baby that's also fertile, then you define these two populations as just different species. That's our best definition of it. And the way they arise, Ernst Mayr, the great biologist, what he developed is this concept of a kind of geographical isolation that leads to this lack of communication between two populations. So if you have a population, okay, and everybody can mate with everybody else, but now like a river will come through, so now the populations are split. And if now, for a long time, no individual from here mates with an individual from there, then over time, changes will accumulate. And to answer your question directly, we don't know, we don't have like a rule of, oh, it's 5 mutations and then they can't breed anymore. It's not like that. It can actually be even just an individual change can already lead to a new species, because an individual change could actually be very large. An individual change could be the copy of an entire part of a chromosome or even an entire organism, and that would already set the two populations apart. So we don't have a hard and fast rule. Yeah.
Yeah, sure. So, I mean, just to get a little bit more into the complexity of how our genome works, how is it that as a species, within our species, for example, humans, we share 99.9% of our genes among individuals, but we still can have such phenotypic diversity?
Right. Yeah, because you and I are 99.9% identical at the DNA level, and yet we may look a little bit different. We may have different heights. We may have different eye colors. I would say, we could focus on the stuff that is the same for a second and just say, despite the differences I've just listed, you and I are very similar. We have exactly the same mechanisms for how our eyes communicate with our brain, right? If someone mapped my brain and your brain, they would see striking similarities, like the cerebellum is here for both of us, and the way our gut works, and the operations of the pancreas, and the way our muscles work, all these millions of details are all exactly the same between you and I. So it's, many times the differences between us are very superficial, right? Like exactly what's the eye color. But still, someone could say that, look, even one little change can change something very big. And for example, in fruit flies, geneticists have shown that if you just make one change in a gene, all of a sudden an antenna, sorry, a leg, a leg will come out where the antenna should be in a fly. A whole leg coming out right here with just one little change. So it's not as though there's this proportionality where if you change 20% of the genome, then the phenotype, the way the organism looks, will change by 20%. It's not percent to percent, and the ultimate reason is that there are, again, as we said, the genes are not working one-to-one, but actually the genes are tightly related in this really complex network of interactions. And more specifically, some genes are regulators and some are more sort of like workers. So the lactase gene that I told you about, that's a worker. It does a function, it breaks down lactose. But there are other genes that are regulators. What they do is they turn stuff on, they turn other genes on. And so if you just change the way the regulator works, you can turn off a whole network. So that's why, actually, let's say you go into a room and it's very dark. So you might say, wow, the way this room looks there's many things here, but overall it's very dark. But you could just, with one little change, you could make it very bright, right? You could just flick a switch, if you turn on the light, and now it's like a different room, right? You can see it's a huge change. And the reason is that that switch that you turned is a regulator, and it makes something very big happen, like now there's light. Same thing with genes. You can just flick a switch, and a gene, when you flick it so it's now on, can turn on a huge set of genes and make a big difference. So yeah, it's not one-to-one, and even that 0.1% difference could make a big difference.
Yeah, but when it comes to genetic diversity, is there enough genetic diversity for a concept like race to be scientifically valid?
So the concept of race is a very loaded concept. Some people define the concept of race as a hierarchy of different races. So that this race is below or higher than this race. There's no evidence whatsoever for any kind of hierarchy. No, it doesn't. And we see millions and millions of variants in the population. I think there's 20 million known variants in the human population, and we're constantly finding more. Indeed, every time a baby is born, we have more that we know about. And so there's plenty of genetic variation in the population, and no evidence whatsoever for that definition of race. However, what does exist is the notion of genetic ancestry, which is that if you just give me someone's genome, and I don't know anything about them, I don't know what they look like, I don't know anything, I just have the ACs, Gs, and Ts, then by comparing it to all the other genomes that we've studied so far, we can guess to really surprisingly high accuracy where they come from in the world, what their geographical ancestry is, given their genetic ancestry. So there's a lot of variance. It's very powerful, but I think all of these sociological works have shown us that the concept of race is not only dangerous and hurtful. It's also just wrong, and there's no evidence for it per se genetically.
So, for example, when in medicine we know that people from different ancestries are susceptible to different diseases in different ways, I mean, how should we think about that? Should we think in terms of ancestry, population, for example? What is the term we should use there?
Yeah, I would say geographical ancestry is the right term. I myself, I have a mutation in my FLT4 gene, and because of that mutation, I have a syndrome called Milroy's syndrome, which affects my foot, my ankle, and I think, if you knew my genetic ancestry, my geographical ancestry, you might say, oh, people in this region, 1% of them have this syndrome. So I guess that would help a doctor. But I think what would help a doctor much more, if they're trying to help me, is to have access to my entire genome. They would just look at all of the mutations, and then regardless of comparing to races, they could just see my own mutations as they are, instead of correlates of other things. In other words, I think having the genome now alleviates a lot of the mystery. You don't have to say, oh, in this population, this thing is very popular, this syndrome, because we now know the genetics of that syndrome, and we don't have to guess 40%, 60%. We can just look and see, is that version of the gene present in the individual? And I think that's much more powerful and sort of sidesteps all of the potential kind of hurtful classifications.
Right. So in that case, and with genetic testing becoming more and more accessible over time, do you think that that would make a case for personalized medicine? I mean, in dealing with certain conditions that affect people from certain ancestries?
Absolutely. I think we've had the ability to sequence genomes for quite some time. It's not very expensive. It's about $500 today. But even though that's not very expensive, it's also not very cheap. And still, I think many people would do it if it would really prove itself. And so far, it's been limited. I think that the future is that we will each have our genomes known. And the reason would be because there would be more and more actionable things that you could do about it. At the moment, a lot of it is kind of like nice to know and interesting, and maybe kind of more from like like we said, geographical ancestry point of view. You would see exactly where your ancestors evolved, and that's so cool. But it's not really actionable medically. As more and more medicines come out that specifically take advantage of knowing exactly what version of the gene you have, then it will be more efficacious to get your genome sequenced.
Right. So earlier in the interview, you mentioned the case of CRISPR. Do you think that gene editing tools and techniques can be used to learn more about genetics and how genes work?
Absolutely, we do it all the time. So gene editing and CRISPR is today the most powerful and prevalent way, is used all the time in biomedical labs throughout the world. We use it in my lab. I think it's used in probably most labs that I can think of. It's very common. And the reason it's common is because if you want to learn about a function of a particular gene in a particular process, a very useful thing to do is to mess with it, to change it, to knock it out, to make its expression go higher or lower, or altogether eliminate the gene. That's called a knockout. And or to now take this gene and put it into a different species and study it there. And so we were doing it for many years with other methods. Gene editing with CRISPR has now made it just ever easier. But it's a great tool. in the lab. And just to be clear, I'm talking about, using cells, not people. We're just working in kind of cell cultures and in doing it in people. That's a topic that is very complex ethically, as I'm sure you and your listeners know. And I think a lot more kind of maturity is required of us. It's really challenging us as a species, as a kind of a society. What are we going to do with this powerful, godlike ability?
Yeah, and I think also that earlier we mentioned pleiotropy and perhaps, I mean, even gene editing, we have to be careful because it could be the case that by modifying certain genes to, for example, prevent a particular disease, those same genes could have pleiotropic effects and affect other aspects of health, for example.
That's right. That's right. It's a very complicated code. Uh, my my dad, he likes to talk about it as though it's spaghetti code, because when when programmers write code, it's supposed to be very neat. But one thing that often happens is that over the years, other programmers add parts, and it evolves as well. And it's very difficult to follow. It's like a spaghetti of one thing going to another in a very disorganized way. Same thing, our genome is very disorganized. It's kind of also like a spaghetti code. And so you're absolutely right. You could try to change one thing, and it could lead to very unexpected consequences. And while the motives may be very benevolent, we may try to alleviate suffering and help cure diseases, we could, I inversely generate more suffering because we do it wrong. And so it's a very complicated process.
Mhm. So we've been talking about genetics. What does epigenetics add to the picture? I mean, when we think about how genes work, etc., uh, what do we learn from epigenetics that traditional genetics wouldn't tell us?
Right. So genetics is considering genes as the way that they're written in the DNA code. And as we know from evolution, you need to have variation. And so a variation in DNA is like an A changing to a T. And that variation needs to be heritable. So now the daughters would also, or the sons will also have that T that's been changed. And that change, if it has an effect on fitness, if the individuals that have that T now instead of the A have a different kind of phenotype, and it leads to a change in fitness, then you have to, it's like a law of the universe, you must have evolution, all right? And so just to recap, you needed to have variation, which was heritable, and had a change in fitness. If you meet those three conditions, you must have evolution. And so for many years, we considered changes like this as DNA changes. What the world of epigenetics brings is that now it doesn't have to be changes in DNA. You can have a molecule that just sits on top of the DNA. It's called, one way is methylation, but this is all just jargon. Just think of it as ways that you don't change the actual code, you just sort of put something on top of it. That's the epi. So epigenetics is just on top of the genes. And now, let's think if it's important. Is it a variation? Well, yes, because one gene could have this molecule and one gene may not. So you have variation. Is it heritable? So sometimes these are not, but often they can be heritable. So upon cell division, the two daughters will both inherit the mark on top of that DNA. And can it lead to a change in fitness, we have observed in some cases that these variants are leading to a change in fitness. So even though it's not a change in DNA, it meets the three requirements, this like universal requirement that if you meet these conditions, you must have evolution, which is it's a variant, it's heritable, and it has fitness. So in this respect, epigenetic changes are crucial. Who said that in understanding life, we need to focus on DNA changes? It could be that the way genes replicate also involves these epigenetic changes. So it's fundamental stuff, and it's very exciting, mainly because we now have the tools to go after it and really measure it carefully. So to answer your question, very important.
Can approaches like, for example, proteomics and transcriptomics give us new insights into gene expression?
Yeah, absolutely. So what these methods have in common is the omics at the end, right? Proteomics or transcriptomics. And what this means is that they measure not just a particular entity, not just a particular protein or a particular transcript of a gene, but everything. And so it's giving you a complete picture of what all the transcripts in a cell are doing. And if you know what all the transcripts, which transcripts are occurring, you know which genes are being transcribed. If you know which proteins are there, you know exactly what's in the cytoplasm right now. And also, as important, what is not in the cytoplasm right now. And this kind of complete picture is so crucial, particularly when you compare it over different dimensions. For example, you compare it over time and see, for example, throughout development, which genes are coming on and what's coming off, and in terms of the transcripts that are made or the proteins. And, in my mind, the most profound aspect of this is that it really leads to the ability to explore and find things that you may not have had a hypothesis about initially, because you are collecting this kind of this data set, this omics data set, and you may not have a specific hypothesis. And even if you do have a specific hypothesis, you're also getting so much more information, and you can just explore it. And if you plot it in particular ways, if you do careful analyses and look at it from this direction, from that direction, try all these ways to analyze the data. The computational biology here is actually really important because it's so much data, so you need to have computational skills. If you do it, you can really find new things that we may not have even had a clue about, so we couldn't have hypothesized about. You just see it. So that's one reason why I really love proteomics and transcriptomics, is this kind of like unbiased, systematic view, allowing us to create new ideas.
Right. Okay, so just one last question. What do you think are some of the most interesting questions in genetics that are not answered yet?
Yeah, I think if you just walk into any hospital in the world, I think you'll see a lot of suffering. So as long as people are suffering, it's usually the case that we just don't understand something. So I think solving diseases is a very important goal of genetics. And by collecting genetic data, by correlating which patients have these versions of the genes and which don't, we can really map out the causative genes, we can map out the pathways, we can understand the biology, and by understanding the biology, we can make a cure. And if anyone ever tells you that we know everything about genetics, just ask them to go to a hospital. And I think it's immediate evidence that there is so much to still understand. And so if young people are listening, but I want them to really appreciate this point, that it is not the case that everything has already been discovered. Sometimes we scientists, we kind of highlight everything that we know, and a lot of our discussion today is about stuff that we already know. But perhaps what we should highlight, and that's what you're getting at with this question, is everything that we don't know. And I think that part is so vast, and it's much more than there is to know. So the young people should, I think, feel as though there is so much to do in genetics. For example, diseases. Another is genetic engineering, because there's so much that we can do to create more nutritious food. For example, if we engineer crops in a particular way, and I know that in some places this is a controversial topic, but it's perhaps as controversial in the same way that vaccines for the coronavirus is controversial. In other words, not so much for a scientific reason. I think scientifically, to create better crops is such a worthy goal. And we don't really know how to do that. I think for many engineering purposes, we just need to have better biology known, and genetics is such a great way of studying it. Other than that, I think that the basic science of, as you mentioned, cancer biology, developmental biology, neuroscience is so exciting. We still don't know what is a thought. How is a thought stored in the brain? We really are just starting to learn. So there's many directions in biology, and it's a very exciting time as ever to come into it.
Great. Okay, so apart from the book, which is again The Society of Genes, and I will be leaving a link to it in the description box of the interview, where can people find your work on the internet?
I don't think I'm very hard to find. I think on our lab website, we show a list of all our publications. In particular now, Martin and I are working on a new project called Night Science, and listeners can hear more about that in our podcast called the Night Science Podcast. And also, there's a series of articles that we publish in Genome Biology about night science. And what this is about is how it is that the creative process works in science. I think it's something that's not taught. A lot of young people that are seeking something creative to do in life sometimes think that science is not creative. And if they want to do something creative, maybe they should go into the arts. I think that's completely untrue. In science, things are as creative as in the arts, I would argue. There is this amazing contribution of creativity that's not well taught, and that's what Martin and I are exploring.
Okay, great. I will be leaving also links to that in the description box of the interview. And Dr. Yanai, thank you so much for taking the time to come on the show. It's been a pleasure to talk to you.
Thank you, Ricardo. It was a pleasure to talk to you.
Hi guys, thank you for watching this interview until the end. If you like what I'm doing, please do not forget to support the channel. You can go over to Patreon at patreon.com/the Dissenter. And you also have links to PayPal in the description box of the interview. Otherwise, and if you like what I'm doing, please share the interview, leave a like, leave a comment, and hit the subscription button. This show is brought to you by Enlights, Learning and Development Done Differently. Check their website at enlights.com. I would also like to give a huge thank you to my main patrons and supporters: Karen Litske and Blanchett, Perga Larsen, Laguerrero, Francis Fordens, Frederick Sunda, Ricardo Vladimiro, Craig Healy, Adam Castle, Olaf Alex, Jonathan Wissel, Jacob Klinkby, Matthew Whitting, Bernardo Wolf, Tim Hollacy, Eric Alenia, John Connors, Paulina Barron, Philip Force Connolly, Jerry Mueller, Herbert Gintis, Rutger Voss, Bo Weingard, Rebecca Newberger Goldstein, Dan Demetriou, Robert Windager, Rui Inácio, Arthur Coe, Zoop, Marco Neves, Colin Holbrook, Susan Pinker, Thomas Trumbull, Bernardo Seixas, Pablo Santurbano, Simon Columba, Jorge Spinha, Phil Kavanagh, Corey Clark, Mark Blythe, Roberto Inguanzo, Mikkel Stormir, Eric Neumann, Samuel Andreev, Tiago Nunes, Bernard Hugni, Alexander Dunbauer, Omari Hickson, Fergal Cusson, Yevon Bodrenko, Hal Herzog, Don Ross, Jonathan Librant, Oslam Bullitt, Nathan Nguyen, Stanton T, Samuel Correa, Eric Hines, Mark Smith, J.W. João Eira, Tom Hummel, David Sloan Wilson, Yassila Dez Araújo, Ethan Solon, Romain Roach, Dmitry Grigoriev, Diego Londonho Correa, Tom Roth, Yannick Punter, Adana Rusmani, Charlotte Bliss, Nicole Barbaro, Adam Hunt, Pavel Ostaszewski, Nelek Bach, Catherine and Patrick Tobin, Al Ortiz, Guy Madison, Gary G. Elman, João Linhares, Lida Cosmides, Saima Afzal, Adrian Yegi, Nick Golden, Paulo Tolentino, João Barbosa. My producers, Isar Webb, Jim Frank, Lucas Taffini, Akian Gilligan, Sérgio Quadriano, Luís Caetano, Tom Van Agdam, Curtis Dixon, João Linhares, Benedict Mueller, Vega Guidi, Sardas France, and Nirban Balachandran, and my executive producers, Michel Rugieski, Rosie, James Pratt, and Matthew Lavender. Thank you for all.